Hello everyone, welcome to today's lecture. So yesterday we finished with the different components of a cell. Okay, there are two types of cell for this year, plant cell and animal cell. We studied that the different components of the cell is first is cell wall, then plasma membrane or cell membrane, then cytoplasm and then cell organelles. Today we will be going to study about the different structure and function of the different cell organelles which are present in the cell. So in the other lecture we had studied there are two types of cell organelles, one which is single membrane bound and the other is double membrane bound. Okay, single membrane bound means it is bound only by a single covering, that is membrane, and double membrane means it is bounded by a double membrane. Okay, it is covered by a double membrane. The first cell organelle which is there in our syllabus is nucleus. Okay, on page number 69 they have given an activity. You have to take a drop of water on a clean glass slide. Using an ice cream spoon, gently scrape the inner surface of your cheek and you have to place it on the slide. You have to put the stain. Stain will give color to the cell and then you have to observe it under the microscope. Okay, you may see in the middle of the cell. Okay, generally it is in the middle of the cell. There will be the nucleus. You might be having these practicals in your syllabus. Now, what is the structure of the nucleus? On page number 70, they have given a diagram, figure 10.7. Okay, they have labeled some parts of the nucleus. I have given you a different diagram so that you will understand. Now, I had told you that nucleus is a double membraned structure. Okay, double membrane structure. That means its covering is double membrane. There are two lines. You might see there are two lines which I have drawn. It is a double membraned structure. Okay, in between there are small pores. Okay, there are small openings which are called as pores for the entry or going out of substances in and out of the nucleus. Inside there are thread-like chromatin fibers. Now these chromatin fibers are basically made up of DNA. You might have heard about DNA. DNA is a nucleic acid. It makes the chromosome. Okay, so DNA makes the chromosome. So DNA is forming into chromatin and chromatin get condensed to form chromosomes. So these are chromatin structures which are present inside the nucleus. There is a matrix inside which is called as nucleoplasm. There is a dark colored structure inside the nucleus which is called as nucleolus. So these are the four things you have to study about in your syllabus about the structure of nucleus, that is nuclear membrane, nuclear pore, nucleolus and chromatin fibers. Okay, so now the functions. What are the different functions of the nucleus? The first function is controls all the metabolic activities of the cell. What do you mean by metabolic activities? Metabolic activities means the different reactions which are taking place inside the cell. For example, photosynthesis is a chemical reaction. Respiration is a chemical reaction. Okay, so there are several different chemical reactions which are taking place inside the cell. Okay, and who controls these reactions? The control is in the hands of nucleus, and inside the nucleus who is responsible? It is the DNA or the chromatin which is present inside the cell. So what is the first function? The first function is it controls all the metabolic activities. Again, what are metabolic activities? The chemical reactions which are taking place inside the cell. The second function is transmission of hereditary characters. You might have heard that hereditary characters means the characteristics which are transferred from parents to young ones, to the next generation. And through what? What is the messenger? Who carries the information? The DNA carries the information. Where is the DNA present? The DNA is present in the nucleus. Okay, so what is the function? Second function of nucleus, it is transmission of hereditary characters. Hereditary means which is transferred from one generation to the next. Now you may see on page number 70, do you know? First point, due to loss of nucleus, large quantity of hemoglobin can be accommodated in the RBC and thereby large amount of oxygen can be transported. Now what is RBC? RBC is the blood cell. Okay, in our blood there are three types of cells: RBC, WBC and platelets. RBC, what are the functions of RBC? First of all, it gives color to the blood. Second, it helps in transport of oxygen. Okay, it helps in transport of oxygen. Now in these RBC there is no nucleus. RBC is a cell which is present in our blood. In those RBCs there is no nucleus. Why there is no nucleus? So that it can carry more amount of oxygen. Next, due to loss of nucleus of sieve tubes of the plant phloem, they become hollow and thereby transportation of food becomes easy. Now what is sieve tube? Sieve tube is a type of phloem. It is a part of phloem. What is phloem? Phloem is the vessel in a plant which help in transport of food materials. In plants also there is transportation of food and water. In animals also there is transportation of food and water. In our case, okay, who is transporting food and water? It is blood which is transporting food, water and oxygen. Okay, now in case of plants, who transports? It is xylem and phloem. Xylem is responsible for transport of water and phloem is responsible for transport of food. Now this phloem, in this phloem there are four types. Out of those four types, one is sieve tubes. In those sieve tubes there is no nucleus so that it can transport more amount of food. Okay, now the next is endoplasmic reticulum. Okay, now endoplasmic reticulum, again I have given a different diagram. In the textbook they have given a different diagram. It is just for you to understand. Now endoplasmic reticulum is a cell organelle which extends, which connects the plasma membrane to the nucleus. It extends from the plasma membrane to the nucleus and they are interconnected. They are interconnected to each other. So what is the main function of endoplasmic reticulum? It is conducting of substances. It connects plasma membrane to the nucleus. That forms a network. It is a net-like structure consisting of interconnected tubes filled with a fluid. It is a network of interconnected tubes and it extends from the plasma membrane to the nucleus and it keeps the cell intact. Okay, next is the functions. It forms the framework of the cell, the framework of the cell. Just like we are having bone as a skeleton, just like that endoplasmic reticulum forms the framework of the cell. The second is conduction of proteins. It helps in conduction of proteins. Third, toxins are made water soluble. Now what are toxins? Toxins are poisonous substances which are formed inside the cell due to several chemical reactions, or if we are eating some food which is poisonous, consider, or it is having some kind of poisonous material, that poisonous material is taken up by the endoplasmic reticulum. It is made water soluble and it is flushed out of the body. So it helps us from getting poisoned. Okay, now the next is Golgi complex. Okay, now page number 70, there is a think about question. Which materials are used to pack your favorite biscuits and chocolates? It is paper, plastic, foil, etc. You might have observed. So it is used for packaging of food materials. Next, what is the function of packaging department? Okay, the function of packaging department is to give a physical barrier to the food product so that it doesn't get destroyed or damaged due to transport or due to storage. Okay, so that is the function of the package department. So in our body also, whatever proteins are produced, it has to be packed and stored inside the cell. Okay, whenever we need it, it could be used. Now this packaging is done by this cell organelle, that is Golgi complex. So Golgi complex is the packaging department of the cell. What is the structure? It consists of several sacs which are placed parallel to each other. These sacs are in contact with each other. They are in contact with each other. They are having openings. Okay, now whatever protein is produced inside the cytoplasm, that protein it is having a rough covering. It will come through this phase, that is cis formation phase. Now it can first we will see the structure. It consists of 5 to 8 hollow sacs which is called as cisternae. This is a very important objective question, that is cisternae. These single structures, okay, single sac, it is called as cisternae. It is hollow sac. The proteins come through the cytoplasm. Where are the proteins formed? The proteins are conducted by the endoplasmic reticulum. From the endoplasmic reticulum, it is in the cytoplasm. Through the cytoplasm, it comes through this phase to this phase, that is the cis phase. Cis phase is also called as formation phase. You may see they are in the form of small globules called as vesicles. Those vesicles will enter into the Golgi complex and it will move like this. It will move like this through each of sacs. When it is moving, it is getting a covering. It is getting packaged. Okay, it is getting packed. So it is modified with enzymes. Enzymes means they are juices which will protect the proteins, okay, which will protect the proteins from damage. And enzymes, it is packed in intact vesicles. Vesicles means small globules, bubbles you can say. So packed in vesicles, and it comes out through the trans phase. It comes out through the trans phase. Okay, so this is the movement of proteins through the Golgi complex. I repeat, it will enter the in the cell. The proteins are formed in the endoplasm. They are come through the endoplasmic reticulum. From the endoplasmic reticulum, it enters the cytoplasm. From the cytoplasm, it will enter the Golgi complex through the formation phase. It will move through the cisternae. When it moves through the cisternae, it is getting packed. Okay, it is getting modified with the help of enzymes, and it will form intact vesicles, and it comes out through the trans phase or the maturation phase. Okay, that will it will exit through from the Golgi complex. So what are the functions? The first function is it is the secretory organ. It is not secretary, okay, or secretory. It is secretory organ. That means it secretes something. What does it secrete? It secretes proteins, the intact proteins, properly packaged, packed proteins. So that is called as secretory organ. Second is it modifies, sorts and packs the different materials of proteins. Third, it produces vacuoles and secretory vesicles. Okay, now these vesicles which are coming out, it is vacuoles or secretory vesicles. It is proteins basically. It helps in formation of cell wall, plasma membrane and lysosomes, because the formation of the cell wall, cell membrane all require proteins or other materials. All these are formed in different parts of the cell, and it is packaged in the Golgi complex. That is why it is called the packaging department of the cell. Okay, so this is about nucleus, endoplasmic reticulum and Golgi complex. Now we will see the next. Now we will see the next organelle, that is lysosomes. Lysosomes are called the demolition squad or the suicide bag. Okay, you will be getting questions, objective like questions in what is the demolition squad of the cell. Okay, what is known as the demolition squad of the cell, or which organelle is called the suicide bag of the cell. Such questions could be asked. Okay, so for that you should be knowing these names. Demolition squad is lysosome, suicide bag is lysosome. Okay, now it is simple single membrane. Okay, it is bound by a single membrane. It is filled with digestive enzymes. What are enzymes? Enzymes are juices. Okay, you can consider it as chemicals which increase rate of reaction. Okay, which increases the rate of chemical reactions which are taking place inside the cell. Okay, so lysosomes are simple single membrane filled with digestive enzymes. Now the functions. First, immune system. In the immune system, it destroys the virus and bacteria with the help of the chemicals which are present inside the lysosomes. Okay, so killing of the viruses and bacteria in our body is done by immune system, and that immune system basically functions with the help of lysosomes. Okay, next is demolition squad. It demolishes, destroys worn out cell organelles. Worn out cell organelles means whatever cells are formed today, it won't be lifelong. It is having a particular life period. Okay, after that period it dies. Those died out cells, okay, those dead cells, it has to be removed from the body. You know that we are having dead cells on our skin. When we take bath, the dead cells are removed. Similarly, inside the body also there are some cells which are having a life period. After that life period, those cells die. Those dead cells have to be removed from the body. Those dead cells are digested by this lysosomes. That is why it is called as the demolition squad. So why it is called as the demolition squad? Because it destroys the worn out cell organelles. The next is suicide bag. Once a cell is formed, if the cell is not proper, then it kills itself. You know the meaning of suicide. Okay, now suicide means it is killing itself. When a particular cell is formed, if the cell is not proper, then it kills itself and dies. Okay, inside our body itself. So such condition, that is said to be suicide. So old or damaged cells are burst. They burst out. Why? Because due to the digestion with the enzymes present inside the lysosomes. The digestive enzymes which are present inside the lysosomes, it will kill the cell. Okay, the cell in which that lysosome is present. That is why it is called as the suicide bag. Next is at the time of starvation. When we are starving, then what happens? The lysosomes digest the stored proteins and fats inside the cell. So these are the functions of lysosomes. Now the next cell organelle is mitochondria. Mitochondria is called as the powerhouse of the cell. Why? Because it produces ATP. What is ATP? ATP is the energy currency of the cell. So we will see mitochondria is called the powerhouse of the cell. It is double membraned. Okay, you may see in the diagram it is having an outer membrane and it is having an inner membrane. The inner membrane is having folds like this. Okay, these are the folds. Inner membrane is having folds. Those folds are called as cristae. This is a very important objective question. The inner membrane, the inner folds of mitochondria is called as cristae. So it is double membrane. The inner membrane is having folds which is called as cristae. Inside that there is a matrix. That matrix contains ribosomes, DNA, phosphate granules, etc. So mitochondria is having its own DNA and its own ribosomes. So that means it can produce proteins on its own. Okay, so it is having ribosomes, DNA and phosphate granules. They oxidize carbohydrates and fats. So now oxidize means it is a type of reaction which you will be studying later. Okay, oxidation reaction. It is a type of reaction. The reactions are carried out on carbohydrates, okay, and carbohydrates and fats to produce ATP. Okay, that means oxidize carbohydrates and fats. Energy is released at the time of those reactions in the form of ATP. The full form of ATP is adenosine triphosphate. Again, an objective question. Functions. What are the functions? The first function is to produce ATP. That is the main function. That is why it is called as the powerhouse of the cell. See, the next function is synthesis of proteins, carbohydrates by making use of those ATP. With the help of ATP, it can produce proteins, carbohydrates, etc. inside the cell. Now the next is vacuole. Vacuole is again single membrane. They are called as storage sacs. They are single membrane structures. Functions. It maintains the osmotic pressure. In case of plant cell, while you drew the plant cell, you might have observed the vacuole in case of plant cell is bigger compared to that of animal cells. So it helps to maintain the osmotic pressure. Osmotic pressure means the water concentration inside and outside the cell. Okay. Second is it stores metabolic byproducts. Metabolic byproducts. What is metabolism? The different chemical reactions taking place inside the cell. During those chemical reactions, there are some byproducts which are produced. Byproducts means some product leaving the main product. Okay, so there is a chemical reaction in which two products are formed. One we need, another we don't need. Okay, that product which we don't need could be considered as a byproduct. Okay, or the products formed, okay, which is not needed now, that could be stored inside the vacuole. Animal cells store waste products and food inside the vacuoles. In case of amoeba, inside the vacuole, food is stored before digestion, and in plant cell, it provides rigidity to the cell. Okay, it provides rigidity to the cell. That means there is enough amount of cell sap inside the vacuole, due to which the cell remains rigid or turgid. That means intact. Okay, so this is about the next three cell organelles, that is lysosomes, mitochondria and vacuoles. Now we will see the next. Now the last cell organelle is plastids. Plastids are double membrane. It gives color to the cell. It gives color to the plant cell. Okay, there are two types of plastids, leucoplasts and chromoplasts. Leucoplasts are colorless or white, and chromoplasts are colored. There are different colors like orange, red, green. Okay, the green colored chromoplast is called as the chloroplast, which contains the pigment chlorophyll, which gives green color to the leaves and the plant, and it helps in photosynthesis. Okay, so the green colored chromoplast is called as chloroplast. It is a double membraned structure. There is an outer membrane and inner membrane. Okay, this is outer membrane, this is inner membrane. Inside there is a matrix, a fluid, which is called as stroma. In those there are coin-like structures which are kept one over the other. Okay, it appears like coins, vada, kept one over the other. They are called as granum. That complete set, it is called as granum, and each coin, the single oval shape, that is said to be a thylakoid. That means this single, only one goal. Okay, one this much is said to be thylakoid, and these three together, it is called as a granum. Okay, so these are the parts of a chloroplast. Now we will see the functions. Inside the chloroplast, inside these granum, the chlorophyll pigment is present, which traps the sunlight. With the help of sunlight, it carries out photosynthesis. Okay, so as it is trapping sunlight, what is the energy which is being trapped? The energy which is being trapped is solar energy. That solar energy, by the process of photosynthesis, it is converted into food. That is chemical energy. Food is a chemical. Which chemical is produced during photosynthesis? It is starch. It is glucose, which is stored in the plant cell. Okay, so solar energy is getting converted into chemical energy. Stroma, it is the fluid which is present inside the chloroplast. It contains enzymes, DNA, ribosomes, carbohydrates. These are all required for photosynthesis. What are the functions? The first function is to convert solar energy into chemical energy. Solar energy is with the help of chlorophyll, it traps the sunlight, and that solar energy and carbohydrate, it is getting converted into carbohydrate. Okay, what are the chemicals which take part in formation of food? It is carbon dioxide and water. With the help of sunlight, carbon dioxide, water and chlorophyll, okay, it the plant forms food product, that is glucose. That is photosynthesis. So solar energy is getting converted into chemical energy. It gives colors to the flowers and fruits. Leucoplasts, which are white colored plastids, it helps in storage and synthesis of food, starch, oil and proteins. Now you may see the textbook. Now in mitochondria and this chloroplast, the plastids, they have their own DNA, okay, and they are both double membrane. There is a statement, the second last paragraph in the textbook on page number 73. Mitochondria and plastids can produce proteins as well as they can replicate themselves as they have their own DNA and ribosomes. It is a very important point. So these are the different cell organelles. You have to study the structure, the diagram and the functions of each and every type of cell organelle. Any of them could be asked. The most frequently asked one is nucleus, okay, nucleus, lysosomes, mitochondria and plastids. Okay, from the other parts generally they ask give reasons. And the diagram of mitochondria, chloroplast, nucleus, these are the frequently asked questions. Okay, now on page number 74 there is a distinguish between prokaryotic cell and eukaryotic cell. Now what is prokaryotic cell? A prokaryotic cell do not have nucleus. Okay, it does not have the nuclear membrane, but it has a chromosome. Okay, that means it has DNA, but in case of eukaryotic cell, it has a proper nucleus. We may see page number 74, eukaryotic cell, the size is about 5 to 100 micrometer. In case of prokaryotic cell, it is 1 to 10 micrometer. Eukaryotic cell, the number of chromosome is more than 1. Prokaryotic cell, there is only 1 chromosome. In eukaryotic cell, the nucleus is with nuclear membrane, nucleolus and nucleoplasm. What is nucleoplasm? It is the fluid inside the nucleus. Prokaryotic cell, nucleoid resembling nucleus. What is nucleoid? Nucleoid is just the chromosome present inside the cytoplasm. Mitochondria and plastids are present in eukaryotic cell. In prokaryotic cell, they don't have membrane bound cell organelles. In eukaryotic cell, example, present in highly evolved unicellular and multicellular plants and animals, and in case of prokaryotic cell, the example is bacteria. This is a 2-mark question. Distinguish between prokaryotic cell and eukaryotic cell. So we have finished with the first chapter of term 2. Okay, it is a very important chapter. In this chapter you have to learn the diagrams, very important. Okay, then the functions and structure of different cell organelles. Okay, you may go through the textbook. If you are having any doubt, you may ask. Thank you.